So welcome everyone once again. Premas is going to contribute with respect to the next generation omics technology and its applications. And what we are going to talk basically, the 2 semester courses, the course 1, which is all about introduction to non-clinical NGS application and genomics analysis tools. Course 2, which will be a part of second semester, where we are going to talk about protocol for clinical next generation sequencing and its application, followed by a campus immersion program, which is a hands-on training where all the participants are going to learn starting from the DNA, the basic material, the process of the library preparation up to the data generation part and the analysis of that data. When you talk about the course 1, where the participants are going to learn the foundational knowledge in NGS, we will be introducing the various genomic analysis tools and techniques, and it will be a 36 hours or more than that of weekend classes. Course 2 will be focused on the comprehensive NGS clinical portfolio, that how you perform experiments in a hospital and a clinical setup. We will be providing examples of real world clinical diagnostics and then 36 hours of weekend classes. Why we are doing that? Because Premas is committed to advancing genomics education through hands-on training. That's why the expert-led courses has been designed, and we are also providing opportunities for the internship if you are able to perform well in these programs. Coming back to the course 1, which is all about the fundamental of NGS library construction. So you will learn from the DNA how you proceed with the various library preparation approaches. What is the difference between a non-clinical and a clinical library preparation protocol? What are the specialized NGS techniques? How the NGS experiments will be planned? And if you're a researcher or you're working in a clinical setup, how will you plan your NGS experiments? And what are the onboard instrument analysis tool available with the Illumina platform? Course 2 will be all about emerging technologies in the clinical genomics space, all infectious and non-infectious samples, how you proceed with the clinical library preparation, and the best possible way to perform the clinical genomics experiment. All of you have already seen the process of NGS library preparation, that you start with a basic sample. You go ahead with the different library preparation protocol. You use a sequencing machine, any sequencing platform from Illumina portfolio, and finally you analyze your data to get to the meaningful conclusion. What we believe at Premas is the true development of skill comes from hands-on training, where practical application turns theoretical knowledge into real-world expertise. And we strongly believe on that idea. What are the sequencing applications that you are going to learn? Coming back from any background, be it microbial, plant, or the human biosciences background, anything related to the library preparation on genomics experiment, we are going to talk about the DNA part, where it could be the targeted. We are going to talk about the exome panel or the amplicon-based sequencing method. When you talk about the whole genome, we are going to talk about the human genome or resequencing, or de novo, where we are going to talk about mitochondrial and metagenomics approach. In the regulation part, we will talk about the protein, RNA, and DNA. When you talk about the protein, we'll talk about RNA binding and transcription factor binding part. For the RNA, we'll talk about the mRNA, transcriptome, small RNA, or miRNA sequencing approaches. Also, we'll focus on the epigenetic part when it comes to the gene regulation process. This is just a summary of what we are going to talk. There are various nuances. We are going to talk multiple aspects of next generation sequencing, so that you will be enabled to plan your research experiments, plan your clinical setup experiment. When you're working in a clinical setup, you will be able to follow the SOP or the mandate of that lab itself. This is just a brief summary of the campus immersion program, which was the part of the second semester course. We have conducted the workshop, the campus immersion itself, on the bacterial whole genome sequencing and HLA typing. Each of the participants got an opportunity to prepare their own library, and then the QC processes involved in those library. And finally, we have pooled the library and loaded it onto the sequencer. And we were able to conduct the two different approaches, that is bacterial whole genome sequencing and HLA typing. This is just a glimpse of whatever the participants have learned on those campus immersion program. And the future plans that we offer at Premas is that if you are able to perform better in these programs, we will be able to provide you the further advanced training opportunity for the selected participant. We will be able to provide you or train more with respect to the hands-on experience in cutting-edge NGS technology and application. And you will get trained at Premas by their field application specialists and scientists. Also, we'll look into the potential career opportunity at Premas upon successful completion. And what we believe is the nurturing the next generation of genomics professional who is having the real world skills and knowledge. Thank you.